Good afternoon, everyone. Welcome back from your lunch break and to an exciting session. And we have heard before a lot about genetic modifiers and somatic instability, and this session is about this specific topic. My name is Nguyen. I'm a clinical geneticist, and I've been working on HD for more than 20 years, with mainly also genetic modifiers. And
I'm Mahmoud Pouladi. I'm an associate professor of medical genetics at UBC in Vancouver, and I've also been working on Huntington disease since 2003 with interest in preclinical modeling and therapeutic development.
So it's our great pleasure to be sharing this session, and we would like to start with the first speaker. And we are very happy that Bob Handsaker from the McCarroll Lab at Harvard University Medical School could come today and speak to us. And his research focused on structural variation in the human genome and its role in evolution and disease. And Bob has worked on the analysis of structural variation from short-read sequencing data. He's also the author of the Genome Strip software, and his talk today is titled The Ticking DNA Clock, Somatic Expansion of the Huntingtin CAG Repeat and its Biological Sequelae. Please, Bob, the floor is yours.
Thank you, and good afternoon. I am Bob Handsaker, and thank you to the organizers for the opportunity to talk to you today. I'm going to tell you about recent work in Steve McCarroll's lab, where I work, and this is work in conjunction with Sabina Beretta's lab, our close collaborator, about somatic instability and how it affects Huntington's disease. So I just have one disclosure. My employer has filed some patent applications related to portions of this work. So our motivation for this work is to try to understand the pathology of Huntington's disease at the neuronal level. If we can understand the underlying cellular pathology in greater detail, this has the potential to guide therapeutic development, both the development of the current therapeutic approaches and possibly suggesting some potent new approaches, for example, some of the things Sarah Tabrizi talked about before lunch. We've known for decades that the CAG repeat in HTT exon 1 is somatically unstable. Early studies revealed that not only is the repeat unstable, but the degree of instability varies across tissues. Small pool PCR experiments from Peggy Shelburne showed that some cells exhibit extremely long expansions, up to 1,000 CAGs. And the GWAS studies that searched for modifiers of the timing of HD onset demonstrated multiple associations to genes that encode for DNA maintenance proteins, implicating these DNA processes and therefore quite possibly somatic instability in HD pathology. Functional studies were then done that linked these DNA maintenance proteins to somatic expansion in vivo, suggesting that somatic expansion may be the mechanism by which these common variants were affecting age at onset. So somatic repeat expansion has long been associated with Huntington's disease, but a key question has been whether somatic expansion drives HD pathology, and if so, how and when. So to investigate this, we collaborated with Sabina Beretta, as I said, at Harvard Medical School and McLean Hospital. We wanted to analyze postmortem brain tissue from HD patients and controls using single-nucleus RNA sequencing. One of the earliest single-cell technologies, called DropSeq, was actually developed by Evan McCosco when he was a postdoc in the McCarroll lab around 2013 to 2015. But we faced a challenge in this conceived work. Standard single-cell sequencing doesn't tell you the length of the repeat in each cell, but that's what we wanted to know. We wanted to know how the repeat was affecting the biology of the cell. So we needed a way to measure the length of the CAG repeat in individual cells alongside those cells' wider RNA expression. So we developed a new approach to do exactly that. And we do this by making two sequencing libraries from each set of barcoded nuclei. One library is a transcriptome library that tells us which genes a cell is expressing. And the other is what we call an HTT CAG library, which gives us the sequence of the CAG repeat from the HTT transcripts. We sequence the HTT CAG library using long PacBio reads, and this allows us to accurately sequence the repeat, even if it is greatly expanded. Importantly, the sequenced repeat contains the cell barcode, which allows us to relate the CAG repeat sequence back to the RNA expression profile of the cell it came from. This new approach allows us to study somatic repeat expansion and its direct biological effects in ways that were not previously possible. So armed with this new technology, we began to look at HD at the single cell level. And we focused initially on the caudate, where we first looked at cells that were at which cells were lost and which cells were not lost in different people in different stages of disease. On the left, these colored bars are showing the proportion of different types of cells in the caudate of controls on the top and people with HD on the bottom. The people with HD are ordered by their CAP score. The CAP score is based on the product of their age and their inherited CAG repeat length, which is a pretty good proxy for disease progression. And as you can see, there's a shift in the representation of the cells as you go down to the HD cases. On the right, you can see that in controls, about 45% of the cells in the caudate are SPNs, and the SPNs are lost in people with HD, and these losses increase with increasing CAP score. From this new data, we were able to see that somatic expansion is exquisitely cell type specific. As shown here in the caudate data from one donor, we see profound somatic expansion of the CAG repeat and the SPNs. These are the striatal projection neurons. And these are on the far right. And we see tremendous somatic expansion there, but only incremental instability in the other cell types in the caudate. And we found this to be true not just in one donor, but in every donor we looked at. The long somatic expansions occurred only in the vulnerable cell types that are lost in HD. Our new assay allows us to see extremely long CAG repeat expansions, previously not detectable at this scale. And we observed some neurons with up to 1,000 CAGs, confirming those early observations from Peggy Shelburne's lab. We also noticed that the length distributions of the repeat expansions had a distinctive shape. While the majority of the cells exhibit only modest expansion, in donors with HD symptoms, there's always a long tail of cells with highly expanded repeats, producing what we call an armadillo-shaped distribution. This big bump on the left, long tail on the right. So we next wanted to understand the temporal dynamics that might drive this pattern of somatic expansion, because we saw these armadillo-shaped distributions in many different donors across a range of disease stages. So we worked on developing some mathematical models and simulations based on realistic biological assumptions that could explain these features that we were seeing. And the models use as input the observed repeat lengths, the age of the donor at sampling, the donor's inherited allele, and an estimate of how many SPNs have been lost based on the cell type composition in the sample. What our modeling suggests is that these armadillo-shaped distributions can arise from two relatively distinct phases of somatic expansion. The first phase is slow. We think it takes many decades for the average neuron to expand from 40 up to about 80 CAGs. But the second phase is fast. After passing 80 to 90 CAGs, the allele expands to hundreds of CAGs in just a few years. On the right, this is a simulation based on the model for a particular donor, showing the predicted distribution of the repeat lengths over the donor's lifetime, along with the predicted fraction of lost cells. Importantly, these models predict that individual neurons enter this rapid expansion phase asynchronously across a long period of time, and they don't even begin to do so until patients are several decades old. So here are the repeat length distributions from 6 donors overlaid and this time plotted as cumulative distributions. You can see that the shapes are very consistent between the donors, but also importantly that there's kind of a kink that occurs between 70 and 90 CAGs in every donor, indicated by the gray area on the plot. This is the transition between the slow and fast phases of expansion. Both phases are well fit by a power law, but the fast phase has about 10 times the growth rate of the slow phase. So this gives us a model for this temporal trajectory of somatic expansion in individual SPNs. Each neuron undergoes a decades-long phase of slow expansion, followed by a rapid expansion phase. But this process is random and asynchronous, with individual neurons reaching phase B at very different times over many, many years. So now I'm going to return to this fundamental question about whether somatic expansion actually drives HD pathology. This is not an easy question. If we do a simple comparison of gene expression data between cases and controls, we see thousands of differentially expressed genes, which are illustrated in these volcano plots. In these plots, upregulated genes are shown in blue if they're significantly uprated, downregulated genes are in orange, and the gray points are non-significant. We see that the differentially expressed genes are found in all of the cell types, not just the vulnerable SPNs. And this may seem surprising, but as we can see in the image on the left, an HD brain is dramatically different from a typical brain. It's undergone tremendous atrophy and neuronal loss. It's had HD for its entire life. The entire brain has been affected, and that's reflected in these gene expression profiles. So the key question is, how can we separate cause from effect at the neuronal level? And we realized we could answer this question in the following way. As we've seen, in any one very tiny tissue sample from a single donor, there are thousands of SPNs present, and they all have different repeat lengths. Some are at 50 CAGs, some are at 80 Gs, some are way up in the hundreds. If we compare these cells to each other, then we control for the shared effects of the donor's life history and disease stage. And this allows us to see the specific effects of somatic expansion acting upon the neuron that has the somatic expanded allele. And this is a very powerful technique. So how does gene expression change if we compare SPNs with low expansion, up to 65 CAGs, to SPNs that have, say, 65 to 150 CAGs? The very surprising answer was that we saw no effect when we compare SPNs in this range of CAG lengths. But what would happen instead if we compared those SPNs on the right tail that are up in the hundreds of CAGs, and we compared, and particular ones over 150 CAGs, and we compared those to the bulk of cells with repeats shorter than 150 CAGs. When we do this, we begin to see hundreds of differentially expressed genes. But these changes only begin to emerge in that long tail of cells with the really long repeat expansions, in particular in the cells with more than about 150 CAGs. So here's a more continuous view of these same gene expression changes. On the top, this is what we call a heat map, where each row is a gene showing distorted expression. Each column in the heat map is the data for a single SPN, ordered from left to right by the repeat length in that cell. In the bottom two panels are the median expression changes of these genes in each cell, with the upregulated genes separated from the downregulated genes. You can see that this trajectory is very stable until a cell reaches about 150 CAGs near the right-hand side of the plot. And moreover, once the changes in expression begin, they intensify with further repeat expansion. And this pattern of gene expression changes was consistent across donors. We call these the continuously changing genes because the gene expression changes intensify with increasing repeat length, but only after the repeat has expanded beyond a threshold of about 150 repeat units. The progressive dysregulation of these genes represents a third phase of SPN pathology at the single cell level, and so we call it phase C. We observed that these dysregulated genes have some specific properties. First, all of the phase C genes are highly expressed in SPNs. Genes that go down in phase C are strong markers of SPN identity, whereas the genes that go up are broadly expressed neuronal genes. The net effect of this is that the phase C changes are a kind of identity softening of the SPNs. And these are the exact same kinds of genetic expression changes that Corinne Marianne talked about yesterday in some of her mouse work, and William Yang is also measuring these same kinds of transcriptional changes in Q140 mice. So this is the third phase of the trajectory of HD pathology in an individual SPN. In this phase, hundreds of genes become dysregulated in a pattern that suggests progressive erosion of cellular identity. We also identified a second group of genes that become dysregulated in the SPNs with long repeats, but they do so in a different way. We call these the phase D genes, and their pattern of dysregulation is quite different from the phase C genes that change expression in a continuous manner. The phase D genes are normally silent in mature SPNs, but we see them start to be expressed as a group in specific SPNs with particularly long repeats. The phase D genes appear to come on together. In cells where we detect expression of some of these genes above background, we tend to see many of these genes expressed. The phase D genes are expressed in SPNs with the longest repeats, but they don't appear to be triggered by a specific repeat length threshold. Instead, the probability of their expression increases as the repeat gets longer. The phase D genes include genes from the Hox gene cluster, genes and transcription factors often expressed in other neuronal cells, but not in SPNs, and also genes CDKN2A and 2B, which are known drivers of apoptosis and senescence. We think of phase D as a kind of de-repression crisis, a short phase of gene dysregulation which is likely followed rapidly by the elimination of the cell. When we began this work, we expected to see toxic effects from mutant Huntingtin protein gradually increasing over a patient's lifetime. But instead, these experiments have led us to a very different model of Huntington's disease pathology. We think the pathology begins with a slow phase lasting decades that's like a slowly ticking DNA clock, proceeding asynchronously in individual neurons. Once a neuron reaches about 80 CAGs, the clock speeds up, and then when the repeat reaches about 150 CAGs, the cell enters a period of gene dysregulation, leading to cell death, likely within a matter of months. And now I want to talk about one last phase in our model of SPN pathology at the single cell level. We call this phase E, when the SPN has been eliminated. We know that there's dramatic loss of SPNs in the later stages of Huntington's disease. Our biological model and computational simulations give us a framework for predicting from a donor's data how many SPNs were in each of these 5 phases over a donor's lifetime. So here's the model for one such donor with a typical inherited allele of 42 CAGs. In the early years of this donor's life, up to their early 20s, the model would predict that almost all of the donor's SPNs would still be in phase A, the slow expansion phase. And as the donor approaches middle age, they would begin to experience slow loss of a few SPNs. But this trajectory of SPN loss would go on to become faster over time. An important implication of this model, especially in the context of therapeutic intervention, is that at any given point in time, the majority of the existing SPNs are still in phase A, the slow expansion phase, and relatively few cells at any given time are in phases C and D, the phases where we think their mutant Huntingtin protein is causing problems. We can also ask what the models would predict relative to milestones like symptom onset. Importantly, we have analyzed just a few donors so far, and in some cases their medical records are pretty sparse. But with those caveats, we might estimate that motor symptom onset tends to fall in the range of about 10% SPN loss, up to 40% or maybe even 50%, although this last number could be biased upwards because of the incomplete medical histories. Our model of asynchronous pathology suggests some surprising answers to some long-standing questions about HD pathology. What is neurotoxic about a typical HTT allele with 36 to 100 CAGs? Perhaps nothing. Perhaps these alleles are not neurotoxic at all. Why does neurodegeneration take decades? We think it doesn't. We think what takes decades is for an individual neuron to reach 150 or more CAGs, but once neurodegeneration starts, it's a relatively rapid process. And why is the degeneration so neuron type specific? We believe this is because somatic expansion rates vary across cell types, and the repeat only reaches this high threshold of 150 CAGs in a few vulnerable cell types. Overall, we estimate that HD pathogenesis is a DNA process for more than 95% of a neuron's life. So the work I've described so far is available as a preprint on bioRxiv. We're also finishing up work on a supplemental website with an interactive web interface which will allow you to explore this data set. And this will be available soon and linked from our lab website. So the preceding work was all from experiments we did in the caudate, but we wanted to see if these results would replicate in brain tissue from the cerebral cortex. We were curious whether the overall pattern would be the same or whether we might observe differences. So in the cortex, as in the caudate, we see that somatic expansion is highly cell type specific. Somatic expansion is most pronounced in the excitatory glutamatergic neurons, although we see a lesser degree of somatic expansion in the inhibitory GABAergic neurons. Very few of these inhibitory neurons tend to reach really long repeat lengths, however, even in late-stage donors. As in the caudate, this pattern replicated across donors and across cortical regions we've sampled, predominantly motor cortex and anterior cingulate cortex to date. When we look at the repeat length distributions in the excitatory cortical projection neurons, we see the same armadillo-shaped distributions we observed in SPNs in the caudate. This suggests that cortical projection neurons also undergo two phases of somatic expansion, slow and then fast. We do see that repeat expansion in the cortex seems to happen more slowly on average than in the caudate. When we look at donors where we've sampled both the caudate and the cortex, we see that the somatic expansion is much further along in the SPNs and the caudate than it is in the cortical projection neurons. In the cortex, we observe both phase C gene dysregulation broadly across the excitatory CPNs, shown here in 4 donors. The threshold for dysregulation appears to be similar to the SPNs in the caudate, again at around 150 CAGs. In the cortex, we see both phase C and phase D type gene expression changes. The phase C genes that change in the cortex are analogous to the phase C genes in the caudate, but they're not the same genes. Recall that the phase C genes in the SPNs were enriched in markers of SPN identity. In the cortex, instead, the phase C genes are enriched in markers of the cortical projection neuron identity, not SPN identity. So these are a different set of genes, but the theme of identity softening is the same. In contrast, the phase D genes that come on during the de-repression crisis are largely the same in the cortex and in the caudate. Importantly, both the superficial and the deep layer neurons appear to be susceptible to the phase D pathology. This is a spatial transcriptomic experiment showing you data from the same tissue section in both plots. On the left, the colors represent the cortical layer of each neuron, and on the right, the colors show which neurons are exhibiting the phase D pathology. And these are in both the deep and the superficial layers. So many key features of the neuronal pathology that we first observed in the caudate are recapitulated in the cortex. There's long somatic expansion, which is highly cell type specific. It starts out slowly with a long, decades-long slow phase, followed by a rapid expansion phase. There are cell-autonomous gene expression changes that emerge in the cells with the very long repeats, and they correspond to both these phase C and phase D gene expression changes. So in the remaining few minutes, I'd also like to tell you about one more set of experiments done by Won-Sok Lee, a very talented postdoc in our lab. Intranuclear protein aggregates are a puzzling and poorly understood feature of Huntington's disease. This is an image of a cell nucleus, and the big blob in the middle is one of these protein aggregates. In mouse models, these aggregates are abundant. They're seen in many, many nuclei. Interestingly, they're also abundant in juvenile Huntington's patients, but in adult HD, they are present in very few cells, even in advanced disease. And this has raised questions about their relationship to HD pathogenesis. After all, how important could they be if so few cells have them? But think about this question in the context of our asynchronous model of HD pathology. The model predicts that any features associated with phases C and D would be seen in only a few cells at any point in time. So we wondered, could nuclear aggregates be such a feature? Won-Sok used some new antibodies to isolate nuclei from postmortem brain tissue that contain these intranuclear protein aggregates. The aggregates are stained pink in this picture, and the nuclei of the cells are in blue. So I'm going to show you the data from one experiment that Won-Sok did in the caudate. In blue is the distribution of repeat lengths in the SPNs from this tissue sample before doing any selection for the cells with the aggregates. The distribution follows the typical armadillo shape, although the long right tail is barely visible in this donor. But after Won-Sok enriches for the cells containing aggregates, he saw a very different repeat length distribution, which is shown on the bottom in orange. The aggregate-containing nuclei, which come from less than 1% of all the cells in the original tissue sample, have extremely long repeat lengths. The mean repeat length in the cells with the aggregates in this experiment was over 300 CAGs. This suggests that these nuclear aggregates are yet another feature of cells that are in the late phase of HD neuronal pathology. Taken together, all of these experiments are suggesting a more detailed understanding of HD cellular pathology. We now believe that the core pathological process is like a ticking DNA clock with slowly accelerating somatic expansion of the CAG repeat. This causes the repeat over decades to eventually expand to hundreds of CAGs in affected neurons. This corresponds then to a long latent phase at both the cellular level and also in terms of symptom manifestation. We don't observe any transcriptional dysregulation occurring at the cellular level until the repeat is in the vulnerable neurons reaches about 150 CAGs. The vulnerable neurons then enter this toxic phase asynchronously. And this is actually very, very important to understanding pathology. The toxic phase, phases C and D, is relatively short-lived, a matter of months, at most a couple of years, for each individual neuron. So if you think about what's happening to a patient 10 or 20 years before symptom onset, only a very few neurons will have entered the toxic phase. And as a result, any phenotypic changes in that patient are going to be very, very subtle, very consistent with the data Sarah showed this morning from the young adult study. And as disease progresses, more neurons are going to begin to enter this toxic phase. But because the toxic phase in each neuron is short, at any given time, only a few % of the surviving neurons are in this phase. And we think this explains, at least in part, why the course of disease can be so long, up to 20 years or more in some of our donors. And finally, the brain is a highly interconnected, tightly regulated organ. All the cells in the brain talk to one another. So we think that many of the manifestations of HD are downstream effects of acute toxicity arising in these very specific populations of vulnerable neurons. So our hope is that these new insights into HD pathology will help to guide ongoing therapeutic development and perhaps lead to some new therapeutic options for people with HD. So what I've shared with you today is the work of a whole team of people in the Beretta and the McCarroll labs. And I want to acknowledge and thank all of them. I also want to acknowledge and thank the CHDI Foundation for generous financial support and for a lot of very deep scientific collaboration. But most of all, I want to end by thanking the brain donors and their families, because without their foresight and their generosity, none of this work would have been possible. Thank you very much.
So we have enough time for many questions because we have one talk less in this session. So please go ahead. Yes, over there.
Hi, I'm right here, Ignacio Muñoz, Factor H. I have a couple questions for you.
Okay.
The first one relates to the isolation of the aggregates. You didn't describe the differential gene expression in those cells. Are those cells also displaying aberrant transcription?
Yes, one talk has gone on to look at that. And as you would expect, because they've got 200 to 500 CAGs, they are exhibiting phase C and a lot of phase D pathology.
Okay, so, sorry, maybe let me rephrase the question again. Do you see a difference between cells that have an expanded CAG without or with aggregates at the level of gene expression?
That's a really good question. And, you know, the sorting is imperfect, right? Some 90% of those, so that the sorting preparations we estimate are like, you know, in excess, like up to 90 to 95% pure, right? And so, and it's possible that that other 5%, right, doesn't, it's just incorrect sorting. On the other hand, when we look for cells that like have aggregates and short CAGs, it's like we just don't see them. If we look for cells with really long CAG expansions and no aggregates, we don't tend to see that. It looks very one-to-one between in this data, in the CAW data, and we've actually replicated this in the cortex. It looks almost one-to-one between the long repeat expansions and the presence of aggregates. It's very difficult to find cells that break that pattern that we're confident in to ask what happens to them.
Thank you.
Up there.
Thank you very much for this really beautiful story, very beautifully illustrated. There was one thing I missed, or at least I didn't understand. did you measure the sizes of the CAG repeats at a single cell? Did you measure them directly from the DNA, or were they inferred from the transcription products, the RNA?
They're inferred from the RNA, which is in the original single-cell protocol is converted to cDNA, and then we're measuring from the cDNA after the 10x library prep.
Yeah, and so the question becomes what dysregulates the transcription, doesn't it?
I mean,
it could be that of course, that the fault is at the DNA level, so the genomic sizes increase. But could it be that the product, the transcription product, that there's a dysregulation which sets in course the whole machinery?
Yeah, I guess in theory it's possible that it's just the transcription product. Although we do, so first of all, there's abundant evidence that there is like somatic expansion at the DNA level, and the fact that we're measuring it at the RNA level, doesn't change that. We also, though, another thing we do is most of the time we only get one HTT transcript from each cell, but sometimes we get two. Sometimes we even get three. And when we get two transcripts, they might be wild type, they might be wild type HD, or they might be HD. When we get two HD transcripts, we get the same CAG length, right? So if it was a transcriptional process, it would have to be somehow, reproducible, that every time it was transcribed, it increased to the same length.
Song has the question he was raising.
There is a question up there first.
Yeah.
Hi, Ruth Luthi Carter. Thanks for your talk. I was wondering if you've considered, so I'm trying to match what I understand from the literature with how you're describing the different phases of pathology. It seems that the phase C sort of cell de-differentiation phenotype, which other people have reported, is sort of the set of gene expression changes that have commonly been observed between mouse models and human, meaning spiny neurons. Would you agree with that?
I think that's generally right. I'm not such an expert on the mouse literature. You can also ask William Yang.
And also, I should point out that some of that pattern of gene expression also is observed in primary cell models where you introduce mutant Huntingtin with the lentiviral expression vector, which is probably not going to be susceptible to the repeat expansion. But that's, again, the phase C, so that still fits with your model. I will follow up on Barry's comment, though, with regard to the phase, I think you're calling it phase C, the transcriptional crisis, or whatever you're saying.
Well, we think...
What is the name for it again, so I get it in my head?
So phase C is the continuous change that sort of starts very mildly at 150 CAGs and then appears to just keep getting stronger as the repeat gets longer. And then phase D is this kind of de-repression crisis where this set of, I'm not a neurobiologist, but these are genes that really shouldn't be on in mature healthy neurons, and they suddenly go on.
So if you go back to Barry's question, because I think he had a really good point that you may want to consider further, the genes that are coming up in the neurons that aren't normally expressed, many of them are actually heat shock proteins, proteasome activator subunits, etc. So I would argue, like Barry suggested, as a mechanism, that it is actually the mutant protein causing protein disfolding crisis in the cell that may be causing the transcriptional de-repression. Thank you.
Song first and then Masih. So.
Hello, this is really fantastic talk, fantastic piece of knowledge. I would like to divert it just a bit. Based on your knowledge, could you comment on the relevance or importance of the CAG targeting in this view as therapeutic strategy?
So, tell me what you mean by CAG targeting, just so I'm answering the right question.
CAG targeting in mRNA to lower the Huntingtin, for example.
So like Huntingtin lowering.
Yeah, Huntingtin lowering by ASO or by siRNA, or specifically targeting the expansions by targeting the CAG mRNA.
Yeah. So, as we've seen, there's lots of clinical trials and studies going on. They're showing early results that are maybe promising for Huntingtin lowering. I think one thing that our research suggests is that maybe that mutant Huntingtin isn't toxic until the CAG gets really, really long. So Huntingtin lowering may be effective at that point in those cells, but it depends a little bit on exactly what the mechanism of toxicity is in phase C and D. Is it mediated through the aggregate? Is it something else about the protein? I think this is not completely understood. Many things are going on at the same time, we think, in phases C and D. And Huntingtin lowering may end up having a beneficial effect through some mechanism we don't understand. So I wouldn't necessarily abandon Huntingtin lowering, but I think there's also plenty of room for other therapeutic approaches. I think, as Sarah said, targeting somatic expansion may be a really potent way to treat disease, and our data suggests that you could maybe do it very early on, and that even once people are symptomatic, you may have an ability to intervene in the trajectory quite strongly, because we think it's quite possible that a lot of the neurons are not in the toxic phase yet, and so you can certainly preserve what's still there.
Now Song, finally.
Thank you, chairs. Hi, Bob. Sung here, CHDI. Thanks for a great talk. I'm over here.
Up there.
Okay.
Can I ask you about the mechanism you propose about cellular de-identification or loss of identity? I was very curious as to whether this is a property of Huntingtin toxicity. And I'm going to ask you something that maybe you're at liberty or may not be, but can you comment on your lab's level of interest or any progress made on the same approach using SCA postmortem brains?
So we haven't looked at SCA postmortem brains. So that's an easy one. What was the first question?
If the mechanism was, you propose the mechanism to be unique to HD, or would you expect it to be a property of the polyQ protein causing this transcriptional de-identification? Thanks.
Yeah, I guess my answer is I think this is an empirical question. I think our work has certainly stimulated interest in looking at the role of somatic expansion and lots of other triplet repeat expansion disorders. Some, I think, historically, we believe there is no somatic expansion. Some we're pretty sure there is. And then the question is, is there a role of somatic expansion, and maybe that should be relooked at carefully in all these diseases, especially one of the important things is that it seems to be exquisitely cell type specific in Huntington's disease. And then maybe it is in these other triple repeat expansion disorders as well. And so you should be making sure you're looking in the right tissue and the right cell type. And then the downstream effects of that somatic expansion might well be different. It's possible that the downstream effects of that somatic expansion do one thing in Huntington's disease and something else in these other diseases.
But you would expect that de-identification can occur in cerebellar Purkinje cells, losing its identity.
In Huntington's?
In SCA.
In SCA.
Possibly. If you're right, it's worth testing, right?
It's worth looking, but I guess, wouldn't be surprised if the downstream effect was different.
Good. There's another question up there.
John Warner from CHDI.
Hello? Hi, John.
Oh, is it on? Yes, it's on. Hello, can you hear me? Yes. Oh, okay, good. John Warner from CHDI. The connection with the aggregates is very interesting because a lot of work is being done to quantify the presence of aggregates in living humans with PET ligands. So that could be a connection that allows turning this insight into a biomarker. That's one comment. But the other comment is, I'm wondering if you've looked a little bit at the composition of the aggregates, because it would seem to me that since they are built up over time, they would probably consist of CAGs of different lengths. and perhaps some information could be obtained by looking at the distribution of CAGs within a given aggregate.
That's a really interesting idea. Yeah, we haven't done that. We haven't done any experiments trying to look at the composition of the aggregates with proteomics or anything. That's not something we've done, but that is a really interesting idea if you could get that aggregate back to its constituents.
Okay, one question here and then Mark up there.
So thanks for the very insightful talk. I was just wondering about the cell type specific of the mechanism of the CAG expansion. Could you elaborate on the molecular mechanism? Why is it more, let's say, pronounced in striatal neurons, meaning spinal neurons, compared to other neuronal types? And what about peripheral tissues? What is known about the CAG expansion there, for instance, liver and others, muscle? Is it also expanded there, and are there also different rates and so on? Can you comment on that?
Yeah, maybe I'll take the second one first because I don't have that much to say about it. We haven't looked in peripheral tissues. I know other people have, Vanessa Wheeler and others. I think our chair's lab has looked in peripheral tissues. My understanding is generally peripheral tissues don't show so little or no somatic expansion except for liver in humans. And then the first question about cell type specificity. Yeah, I think this is a really interesting question, and people who look at the mechanism of somatic expansion are fascinated by this, and I think we don't have an answer yet. But my sense and the way I talk about it is it's exquisitely cell type specific. For example, I didn't talk about this, but we see differences between the direct and the indirect SPNs in our caudate samples. The indirect SPNs expand about 15% faster. So they're very comparable to the DSPNs, and I think we kind of get lucky that we don't have a very heterogeneous cell population, but they are about 15% faster on average. And this is consistent with the idea that so first of all, they're expanding faster, and then they are lost earlier. And in our data, we can see that there's a slightly faster loss to ISPNs than DSPNs.
Good. There's another question, and Mark, then you can go to the microphone.
Great talk, thanks. Question about the cell type specificity as well. Have you looked at potential DNA damage repair marker expression in the SPNs relative to the other cell types? Are they higher or potentially more active?
We have not done that, and we should. Perhaps like the approach might be building a model, and if you were to build a model, right, to try to predict the cell type specificity, probably you would want to include something like HTT expression because we think it's probably transcriptionally coupled. And you'd want to look at the expression levels of all the other players in the mismatch repair complex, MSH2, MSH3, PMS1, FAN1, and then see if there was unexplained variance. It is so exquisitely cell type specific. In many cell types, it's close to zero, right? Maybe in very advanced disease, you see a little bit of somatic expansion. But we actually haven't tried to build such a model yet, but we should. And see if there's some missing components that we don't understand.
Good. Mark, now it's your turn.
Yeah, sorry. One comment about the periphery. I think every time we look, there is instability in the periphery. It's low, very low, but I don't think anybody's found a peripheral tissue where there's none. Thanks, Bob, for the great new updates on cortex and the aggregates. Just a question. Do you know is there any overlap between phase C and phase D genes with other non-repeat expansion neurodegenerative disorders?
So not that I know of. We haven't really looked at. I don't know. I don't know the answer to that one. And Mark makes a good point. I should when I answered the question about somatic instability, I was thinking mostly of large waves. So clearly, the Moncton lab and Mark are looking at using blood as a biomarker. So that's somatic instability in the hematopoietic stem cells, right? Is what's being measured there, and we think this is a biomarker for Huntington's disease. But it's very small. They're measuring like very tiny expansions, like 1 CAG over several years.
Great. Okay, I'll ask maybe the last question, Bob. Terrific talk. So the results when you compare cells with shorter expansions to the ones with longer ones. And you don't see any difference, essentially, right? The 30 to 60 and then the 60 to 150. I mean, these are very surprising because based on a lot of animal models and also stem cell-based models, there is a lot of transcriptional dysregulation, even with shorter repeats. So can you comment on whether there are limitations related to the approach that's used to detect these transcripts that could maybe explain the lack of differential expression?
So I want to say two things. The first is we were shocked by this. We were shocked that we would do different measurements. We'd sometimes see differences, we sometimes wouldn't, and then we realized it was only every time you included the long tail, we saw differences, and when you didn't, the differences weren't there. But what's really, really important is we're not comparing cases to controls, right? If you compare any case to a matched control, you take a Q140 mouse, compare it to a wild type, yes, there are tons of differentially expressed genes. What we're comparing here is two neurons that are sitting right next to each other in the brain, and one has 50 CAGs and one has 100. And we're asking, do those look any different? And the answer is no, even in the HD brain. But if you pick a different neuron that's also right next to those two and it has 200 CAGs, it looks quite different.
Okay, thank you.
Great.